In this video, we will cover what we know about transcription from studying bacteria. Much of what we know about transcription comes from studying the bacterium E. coli. In a lot of ways, this process works the same in prokaryotes and eukaryotes. So let's take a look at transcription in prokaryotes before proceeding to a slightly more complicated version in eukaryotes. An important region of DNA in transcription are the promoters. These are highly conserved sequences just upstream from the site where transcription begins. The role of the promoter is to direct where transcription should begin. If you look at the figure here, you will note that the bases are numbered with negative numbers. This is the typical designation of bases, which are numbered relative to the transcriptional start site, which gets a base number of 1. Promoter regions in bacteria are very conserved, but do have some variation. In bacteria, variation in these promoters affect the rate of transcription, with sequences that vary more in relation to the consensus, resulting in lower rates of transcription. The promoter region is important for initiation of transcription. RNA polymerase, the main workhorse enzyme of RNA synthesis, is made of two components here, the core enzyme and a separate piece called the sigma factor. This holoenzyme binds to DNA loosely until it encounters the promoter region. At this point, the sigma factor recognizes the conserved promoter sequences and begins transcription. So when the RNA polymerase enzyme encounters the first part of the promoter, it forms a closed complex. Once it reaches the -10 region of the sequence, it opens up the DNA to form the open complex. Interestingly, this region is defined by all A and T bonds, which are more easily separated than C to G bonds. At this point, RNA synthesis begins. Once a short strand of RNA is made for the open complex, the sigma factor is released. This marks the end of initiation and the beginning of elongation. During elongation, the RNA polymerase enzyme proceeds down the length of the strand, forming an ever-lengthening RNA strand. The enzyme opens up about 17 bases at a time, and once it passes by, the DNA rebinds back into the double helix. This all happens incredibly fast and is estimated at about 43 nucleotides per second. During elongation, the RNA polymerase slides along the template or the antisense strand in a 3' to 5' direction and uses nucleoside triphosphates as precursors, much like we have seen in DNA replication, just with RNA. Complementary base pairing rules are followed during this process. Within a particular strand of DNA, several genes can be located, and these do not always have the same template strand. The figure here shows 3 adjacent genes with different promoters, which signal the direction of transcription. Notice the direction of transcription changes across the strand of DNA, signaling that genes A and B have different template strands than gene C. RNA synthesis stops at the terminator. At this stage, the RNA polymerase complex dissociates from the DNA molecule. There are 2 different mechanisms which I will briefly mention here. The first is Rho-dependent termination, which requires a protein called Rho to dissociate the complex. A second type of termination is Rho-independent and is facilitated by a uracil-rich sequence located at the 3' end of the RNA sequence. Upstream from this, a stem loop secondary structure forms, which helps pull apart RNA synthesis complex.